This presentation introduces an important genomic technique called GWAS. It looks at its potential impacts on our ability to predict academic attainment. GWAS stands for Genome-Wide Association Study. Each of us has 23 pairs of chromosomes in the nuclei of all of our cells. The diagram shows a small group of bases from part of chromosome 1. One copy of chromosome 1 was inherited from the mother, and the other copy of chromosome 1 was inherited from the father. The bases shown on both chromosomes are similar, except for one pair of bases shown in green. For simplicity, we will only think about the bases on one strand of the DNA on each chromosome. Therefore, the genotype for this person is TC for this particular pair of bases. GWAS studies look for differences in individual bases across a large number of people, or differences in groups of DNA bases. The effects of these individual differences in DNA on a particular complex characteristic is then measured. The kinds of characteristics studied are height, personality characteristics, intelligence, G, and educational attainment. This diagram shows the two chromosomes that make a pair, with one strand of bases shown for each chromosome. You can see that most of the bases are the same. For the base that is highlighted in red, the bases AT are different to other bases in that position in the population. This is an example of a single nucleotide polymorphism, or a SNP. SNPs are also called alleles, or genetic variants. Sometimes we study a small group of DNA bases which are inherited as a single unit. These are called haplotypes. Software companies like 23andMe use reference haplotypes which are known to be linked to ethnicity as a basis for establishing the ancestry of a person. A GWAS study starts by choosing a characteristic for investigation. This could be general intelligence, G, or years of educational attainment, which is sometimes shortened to EDU years. This example will think about human height. Participants for the study give a sample of their DNA, usually from saliva. Saliva can be used because it contains traces of cheek cells, which have nuclei containing DNA. At the same time, a measurement of the person's height is also recorded. The sample of the genome is taken to a sterile laboratory where precautions are taken to prevent cross-contamination of DNA by the research workers. The DNA is extracted from the cheek cells and is copied many times using a machine known as a PCR. This acts like a photocopier for DNA. PCR, incidentally, stands for polymerase chain reaction. It may take several hours for the DNA to be copied. The DNA is cut into small pieces using enzymes. The small pieces are called fragments. The fragments are treated with a chemical dye so that they glow brightly, they fluoresce under ultraviolet light. This is called labeling the DNA. Labeling the DNA helps researchers' computers to find pieces of DNA during the analysis. The DNA fragments are then treated so that the bonds between the nucleotide bases break, and each fragment consists of a single strand of DNA. A key part of a Genoa study is called a gene chip, or more formally, a microarray. A microarray is a glass slide that is printed with thousands of tiny spots at specifically defined positions. Each spot is called a feature. There may be more than 1 million different features on a single microarray. The diagram shows a microarray and a feature. Each feature on a microarray contains a small fragment of single-stranded DNA, which may be 40 to 60 nucleotides long. In other words, it will contain between 40 and 60 DNA bases in a specific sequence. Each individual feature will contain many copies of this identical sequence of DNA. The DNA is attached to the microarray and will not become separated from it during the test. Each feature contains a different fragment of single-stranded DNA. Each DNA fragment on the microarray chip is carefully indexed so that its precise location on the chip is known. The earliest GWAS tests used almost 2 million locations of DNA across the genome to select out those sites which showed useful genetic variation. The participants' DNA fragments are allowed to mix with the DNA sequences fixed to the microarray. Those fragments of the participants' DNA that find a matching base sequence on the feature will bind to it. This is called hybridization. Those DNA fragments that do not hybridize with the DNA held on the microarray are washed off, leaving behind only those fragments which have attached to the DNA on the microarray. These fragments will fluoresce under ultraviolet light, and a computer reads the patterns of fluorescence. It is then able to determine which sequences of DNA variants were present in the participant's DNA. The Manhattan plot combines together the results of all of the participants in the study. The chart is arranged according to the order of the 22 pairs of chromosomes in the genome. The sex chromosomes X and Y are excluded from the analysis. Each dot on the Manhattan plot represents a single DNA variant. Education Attainment, or EA, measures the number of years a participant has spent in full-time education. It's useful because it is collected in many large-scale studies and is a readily available metric. Unlike measures of intelligence like IQ or G, it has been suggested that it is unbiased in the sense that they are not subject to training, faking or anxiety, each of which is a criticism of intelligence tests like WAIS-4, which estimates G. This is a Manhattan plot for a GWAS study of 1.1 million people for educational attainment. The paper identified differences in over 1,200 variants that were statistically significant. These were distributed evenly across the whole genome. The DNA variants in this analysis account for about 11% of the differences or the variance between the individuals in the study for educational attainment. This is remarkable, but it is still some way below the 50% or so that we would predict is the genetic contribution to the differences between people for educational attainment. We expect that if we used a larger sample of people and a screening of the whole genome, then we would probably find the missing variants. The remaining 50% of the differences would be explained by differences caused by environmental factors, reinforcing the key idea that phenotypes like educational attainment arise through interactions between genomes and the environment. Not all of the variants are in or are near protein coding regions of the DNA, and yet they are statistically significant in this analysis. Their functions will doubtless be discovered in the future. However, many variants are associated with nearly 2,000 protein coding genes, and these variants are located in or close to regions of DNA that are expressed during fetal development of the nervous system and also in childhood and adolescence. Many of these variants are associated with genes that encode for proteins that produce nerve cells or neurotransmitters, or activate ion channels, or promote cell signaling or synaptic plasticity. In other words, these are the variants that help to build and maintain brains. Genoa studies of educational attainment show a significant genetic component. As we've seen, the latest version, EA3, predicts about 11% of the differences in educational attainment. This is important because it's now a usable characteristic, rivaling socio-economic status in importance as a research tool. For example, parental EA correlates 0.3 with children's intelligence, accounting 9% of the differences between children. So, how do we calculate a polygenic score for educational attainment? Well, imagine that a Genoa study on an individual finds 10 single nucleotide polymorphisms or 10 variants as shown on the screen. The first step is to work out which of the two alleles has the largest effect on the phenotype. For SNP number 8, allele A has the increased effect on the phenotype, and as there are 2 copies of allele A present, the combined contribution of these alleles is 2. That is the genotypic score for that SNP. For SNP 5, it is the allele G that has the increasing effect on educational attainment, and in this particular case, the person does not have a copy of G, so their genotypic score is 0. When 1 copy of the increasing allele is present, the genotypic score is 1. This is the case for SNPs 1, 3, 4 and so on. Added together, we get a total polygenic score of 9. However, this is weighted by looking at the correlation of each SNP to the phenotype, and then this is used to produce a weighted genotypic score by multiplying the correlation with the genotypic score for each allele. It is clear from these analyses that the effects of any one individual variant is very small. The SNP with the largest effect accounts for only 0.02% of the differences in EA, which is equivalent to about 2 months of education. Polygenic scores for EA are obtained by considering the effects of all the variants in the analysis, even those with low levels of statistical significance. Their effects are aggregated into a single polygenic score, which is called a GPS. Twin and adoption studies predict that about 50% of the differences in IQ scores in a population are due to genetic differences. EA3 predicts only about 10% of the differences in cognitive performance. This difference, which is found in Genoa studies of other characteristics too, has the general term missing heritability. This paper, The New Genetics of Intelligence by Robert Plomin and Sophie von Stum, was originally published in Nature. It is a significant paper because it introduced the idea of polygenic scores to a wider audience beyond the genetics community. It's very readable and very accessible. I suggest that you read it and talk to each other about it. How reliable are polygenic scores for educational attainment as a way of predicting educational success? The diagram shows an early version of educational attainment called EA2, the one that's discussed in the Nature paper. You can see that there is a positive correlation between the polygenic score and the GCSE scores, but there is a considerable amount of individual variation for any one polygenic score. If we look at a polygenic score of 50%, which is midway in the distribution, the majority of people are scoring around about 50% in their GCSEs. However, some people are achieving in excess of 99%, whilst others are scoring only about 3 or 4%. A person with a 50% EA2 polygenic score has the potential to achieve the whole range of GCSE mathematics grades in the 2018 examinations. Even when selecting only for grade 9, the scores range from 15% to 75%. Most of those with the highest EA2 scores did not achieve level 9. These are early days for the technique, and we expect that improvements will be made. Full genome sampling will improve the reliability of the estimates, and this will become a possibility in the next few years as the price of gene sequencing falls. But will it be able to predict more than the 55% of the variance for differences in GCSE math scores predicted by traditional twin studies? In other words, to what extent will it ever be a reliable predictor of educational attainment? What would schools be like if such an accurate predictor was available? This is something that we need to consider from a social context.